At the molecular level, drug discovery is a problem of geometry. The objective is identifying chemical compounds that exhibit high spatial complementarity and binding affinity. To simulate this interaction, medicinal chemists rely on virtual screening. Historically, executing a single screen required passing data through a fragmented pipeline of independent, highly specialized software packages. A researcher might use one tool to format ligand files, another to identify binding sites, and a third to run the docking calculations. Manually moving data between these distinct environments introduces heavy friction and the constant risk of formatting errors. Many of these individual engines rely strictly on command line interfaces. When screening a library of thousands of compounds, the overhead of managing syntax significantly slows down the pace of discovery. The underlying mathematical algorithms are powerful, but accessing them requires a streamlined workflow. Removing this operational friction is as essential to the screening process as the math itself. This operational friction is the specific problem PyRx was built to solve. It acts as a unified environment for the entire virtual screening pipeline. PyRx functions as an orchestration layer, a single graphical interface designed to control a vast ecosystem of established docking tools. Instead of juggling different programs, PyRx routes raw data automatically between a carefully selected stack of engines, handling complex formatting in the background. The developers are explicit that there are no magic buttons in structure-based drug design. A researcher still needs to apply rigorous scientific methodology to configure the screening parameters. PyRx translates raw computational power into a reproducible process, making computer-aided drug design accessible through a single interface. A successful docking run requires pristine data. Before any calculations can begin, the raw input files detailing the candidate ligands must be sanitized. For this first phase of the stack, PyRx integrates an open-source chemical toolbox called OpenBabel. OpenBabel handles the import and standardization of raw SDF files from massive compound libraries. The tool automatically scans the structures and strips out disconnected salt ions that would otherwise interfere with the simulation. Next, OpenBabel runs energy minimization calculations on every imported ligand. This step adjusts the molecule's geometry to its lowest energy state. If a ligand starts the simulation in a highly strained, unnatural configuration, the resulting binding scores will be mathematically flawed. Automating these preparation steps ensures a baseline of quality control, preventing errors from propagating through the rest of the pipeline. With the ligands ready, the software shifts its focus to the other half of the equation, the topographically complex target protein. Forcing an algorithm to search the entire surface area of a target protein for a potential binding site requires a massive expenditure of computational resources. PyRx deploys DeepChem to perform automated binding site detection and focus the search. DeepChem scans the protein and isolates specific physical pockets where a ligand is most likely to attach. This reduces the manual configuration time required from the researcher. Once the pocket is defined, PyRx calls on Autodock tools to map that indentation into a rigid spatial grid. Autodock tools takes those coordinates and generates the exact input files required for the heavy computation phase. Automating target processing allows researchers to scale their screening efforts from individual compounds to entire libraries. We now reach the core of the pipeline. PyRx relies on Autodock 4 and Autodock Vina as its primary mathematical engines. Because PyRx has orchestrated the workflow up to this point, all the formatted data from OpenBabel and DeepChem feeds directly into these engines. Autodock takes over, testing thousands of possible conformations and rotational orientations of the ligand inside the target pocket. For every valid fit, the engine calculates the thermodynamics of the match and assigns a binding affinity score, predicting the strength of the interaction. Utilizing established, peer-reviewed engines like Autodock Vina ensures that the resulting affinity scores are scientifically valid. The user leverages industry-standard mathematical engines without having to manage command-line syntax. Once calculations are complete, the pipeline enters its final phase, translating matrices of output data back into human-readable formats. The application interface and cross-platform functionality are constructed using WX Python and the Nthought tool suite. For structural analysis, PyRx relies on the Visualization Toolkit, or VTK. VTK takes the raw coordinate data and renders the precise 3D spatial interaction between the ligand and the target, allowing chemists to inspect the binding site. Alongside 3D modeling, PyRx utilizes matplotlib to generate 2D plots of the computational output. To manage the volume of results, the software features built-in spreadsheet functionality. All binding affinities are organized into sortable rows and columns. When a screen processes thousands of compounds, researchers sort and filter these spreadsheets to identify candidates with the most promising scores. Coupling background computation with immediate visual feedback allows researchers to iterate on docking parameters and verify results in real time. The ultimate test of this software stack is its track record in active laboratories. Since 2010, PyRx has been cited extensively across a vast bibliography of published biological and pharmacological literature. Researchers frequently use the software to screen natural extracts, discovering bioactive compounds in plants like water hyacinths and citrus peels. It is also used in drug repurposing studies, helping identify existing pharmacological candidates for neurodegenerative conditions like Parkinson's disease and ALS. The platform saw a surge in utility during the COVID-19 pandemic, where laboratories used it to rapidly screen compounds against novel viral targets. Recent studies demonstrate its capability to integrate with broader analytical methodologies, including network pharmacology and the validation of AI-driven molecular models. The fact that researchers from disparate fields utilize the same pipeline validates the stability and flexibility of the architecture. This extensive bibliography illustrates how researchers consistently use PyRx to translate computational models into validated pharmacological leads. Deploying this platform into an active research laboratory is governed by a specific licensing model. The latest supported versions of the software are offered through two tiers, Academic and Pro. There is absolute feature parity between these two versions. Both tiers grant full access to the orchestration layer, including 64-bit support. The Academic tier is discounted by 50%. Both tiers operate on a perpetual license model with no subscription fees. To facilitate high-volume research, the academic license allows users to install the software on an unlimited number of computers within the same laboratory environment. Commercial licenses carry a distribution limit, restricting the software to a maximum of 10 people within a single commercial lab. By unifying complex tools into an accessible interface, this architecture allows researchers to execute rigorous virtual screening directly within their own labs.